Welcome to lesson 6.6. In this lesson, we'll be talking about the plasmodial slime molds. This is another way that soil protists can feed, survive, and produce spores, where the amoeboflagellates mate and produce larger structures. So this is very interesting because it's where the microscopic organisms turn into something we can see and marvel at, a macroscopic, much larger structure. I also want to draw your attention to this beautiful image of Lamprodoma, which has been taken by Sarah Lloyd, our very own Australian self-taught slime mold expert. She has written a book called Where the Slime Mold Creeps, with beautiful images and stories about what she has found. So I highly recommend that book. Okay, so let's begin lesson 6.6. So what are plasmodial or acellular slime molds? These beautiful structures come from either amoeboflagellates or organisms that are actually called amoeboflagellates because they can switch between being an amoeba and being a flagellate. They're also called mixed amoebae. But basically, when the amoebae or the flagellates mate, they actually produce fruiting bodies just like fungi and disperse their spores via those fruiting bodies. A structure that isn't multicellular but grows from the one cell and develops. And there are various ways these structures are developed in various species, but the one thing that they have in common is that there are no multicellular structures formed other than just the one cell developing and growing into a visual structure, visible to the naked eye. In the image here we have Stamonitis, the chocolate tube slime mold, which you'll get to see throughout this lesson in various forms, but just remember that this will become the chocolate tube slime mold. There are three types of slime molds, and the first one we're going to look at is the plasmodial slime molds, also called myxomycetes or myxomycetes. And the mycetes bit or mycetes bit is referring to its fungal-like lifestyle, and that's where the ability to produce spores used to put slime molds into the fungal group of organisms. But they are all distinct organisms that may not even be related to one another but have a similar lifestyle. So a lot of the slime molds are actually far apart in relationship to one another. They come from very different distinct groups of organisms but have adopted that kind of lifestyle and similar life cycles that they all share. An example of that is Physarum, where individual flagellates swarm and fuse into enormous single cell with thousands of diploid nuclei, and we call this giant cell the plasmodium, hence the name plasmodial slime molds. Cold and dried conditions cause the plasmodium to go dormant into a structure called sclerosium, which is an irregular hardened mass of large cysts, also called macrocysts. But when the conditions are right again and or food is exhausted, this structure begins to function again and comes out of its dormancy and sporulates, so forms spores. The mature plasmodium moves to an exposed drier area to form fruiting bodies called sporocarp, and this is also what we call fungal fruiting bodies. And these contain sporangia, where the spores are formed, and also which are the spore dispersal structures. And these serve to disperse the spores of the organisms. The spores then germinate, and from them amoeboid cells called myxamoebae are generated, or flagellated swarm cells might be born from those spores. These organisms can also revert into dormant microcysts when conditions become unfavorable. However, favorable conditions cause fusion of the cells and a single-celled zygote is created, which feeds and grows to form the plasmodium again. So on the right here we have a diagram showing the plasmodial slime mold's life cycle. So it starts when the mature plasmodium, so the growing phase of the large cell that we've been describing, that can form sclerotia when the habitat turns dry. When the conditions are unfavorable and there is no longer any food left, the plasmodium will start to form sporangia. So it changes its shape and begins to form the sporangium, which is made up of a stalk and a structure that is capable of holding and then releasing the spores. What's important to mention here is that this phase of the life cycle, which is the plasmodial phase, is diploid. So that means that there are two sets of chromosomes present, whereas the unicellular life cycle is haploid. So there is just one set of chromosomes present within the organisms, and then they get together and they double up their chromosome number. So when the spores germinate, they undergo a process called meiosis that actually restores that haploid phase where there's only one set of chromosomes, and that's so they can actually mate with others and combine their chromosomes to form a zygote. Once these spores are matured and the sporangium releases these spores, the spores germinate when the conditions are favorable for the germination process, and they can switch between amoeboid or flagellated forms. So they can either become amoebae or they can become flagellates depending on the environment, and they can switch between those two forms. And then when they want to form a plasmodium, they undergo the mating process, which involves plasmogamy, which is the fusion of the cytoplasm of the two haploid cells, which allows them to undergo fertilization, and that's when karyogamy occurs, and that is the fusion of nuclei. And this forms the zygote, which is diploid, so has two sets of chromosomes. And then the young plasmodium starts to feed and grow, and it multiplies its diploid nuclei and eventually becomes a mature plasmodium, which is multinucleated and has a free-flowing mass of protoplasm, and that's how it generates its movement and nutrient distribution within the giant cell that we call plasmodium. And then once again, if conditions become unfavorable, the habitat dries out, they can form a sclerotia to go into a dormant state, or when food runs out, they actually begin to form sporangia and the cycle begins again. So that is it in a nutshell. And now we'll look at some examples of plasmodial slime molds. This time lapse has been borrowed from a YouTube video called Lengths of Time, Slime Lapse by Biographic. So the life cycle we've just discussed is of this particular organism, and this is just the one plasmodium, so one cell spreading forward in the time lapse. Then in the lab it actually looks a little bit different in the sense that it's been trained to absorb nutrients straight from the agar rather than being fed bacteria. So this is one cell just doing all of this work. And we'll talk about the importance of the way it moves, but also notice this pulsating action of the physarum, how it moves back and forth to give this pulsating motion. And this is actually important in its decision-making process, and I'll talk about that next. So slime molds have very interesting ways of being, and one of those is where the movement of slime molds has been studied extensively for our understanding of how these organisms demonstrate cellular movement. And time-lapse shows slow stop and reverse behavior every minute or so, and this creates this pulsation kind of movement where the organism pulsates. And this is caused by a network of fibers and motor proteins, which result in contractions, much like our muscles. And this is thought to be important in decision making as these organisms interact with their environment. The growth of slime molds and the way they advance forward to consume food, particularly, can reach speeds of up to 1.3 millimeters per second or 4 centimeters per hour, depending on the species. So they are, in fact, the fastest microorganisms recorded. And this is showing how Stemonitis actually develops its fruiting body, and then it produces its spores. And you can actually see those spores when you actually interact with the organism. You can actually move it, and there will be a lot of spores dispersed. It's very fun. So if you ever see one in nature, go up to one and have a look if it's at that stage where it's producing spores. But really interestingly, I've found some of them that look like pubic hair when they're really, really dry and just really gone past the sporulating stage. And the leftover hair-like tufts literally look like pubic hair or a beard, something very furry and quite interesting to come upon in a forest. So slime molds are actually incredibly intelligent, and scientists are using slime molds in all kind of experimentation. They actually have spatial memory, so they can actually remember where they've been. For example, when a labyrinth is set up for them, they will find food in the quickest way possible, and the next time that labyrinth is presented again, they will now be able to remember the fastest route, whereas before that they were experimenting as to how to get there. So they have learned behavior transfer, and in fact they've been used to optimize rail systems. For example, the Tokyo's rail system, which spans about 36 different cities, a very big network, when the stations were superimposed with slime mold food, which is oatmeal, when oatmeal has been placed at the various points to mark the various stations of Tokyo's rail system, the slime mold was able to replicate almost exact copy of that system that humans have come up with. In fact, it optimized some areas where there were blockages in the system. So the slime mold had solved and increased the potential of Tokyo's rail system. And this has led to the development of the adaptive network design. It's basically using biological systems to solve very complex networking and distribution problems, which you can imagine takes humans a very long time to plan. Like the Tokyo system would have taken years to design potentially. Whereas if you use biological systems that are actually designed to network perfectly already in nature, that are intelligently already designed, we can actually save a lot of time and come up with solutions to like traffic problems and many other issues that we face. Interestingly, also, NASA has used the slime mold physarum growth pattern to create an algorithm to map the cosmic web, which is made up of dark matter. And so now there is a 3D model of the distribution of the dark matter of the cosmic web based on how slime molds grow. And the various points to demonstrate that were actually where gases were accumulated in the hot spots of the dark matter, and this was used to form this algorithm to show the connections between those hot spots in the cosmic web. So very interesting, and so many different studies exist showing this, and physarum is one of the most well-studied of these. So this image shows the Tokyo subway network, how it's been mapped out by physarum, going from where the physarum was placed and the different stations away from the Tokyo center to the various cities that the subway connects. And of course, these cities are demarcated by its food source. So in this case, oatmeal. And then very rapidly you can see it's moving towards the different food sources. But over time, it finds the shortest routes and fortifies those routes by growing only in between those to gain that food source. So this is how in 26 hours physarum can actually solve the puzzle of the most efficient way to design a railway system or a subway. And then, based on the algorithms that such experiments provide as to the growth pattern, and also in this case it's a 3D model of the cosmic web, so it is not achievable to be viewed in 2D on an agar plate, but if the algorithm is applied, it can be used in a 3D model. In this case, gases that form stars have been used as the points of interest, and voila, we have the cosmic web and the dark matter that it is made up of. What's interesting is that a lot of networks have that same growth pattern, and this is called fractal geometry, based on fractals, where there's continuous division of the various filaments, and this can be seen in all kinds of scenarios, such as, for example, our nervous system. The internet is based on this framework as well. How trees grow, how roots grow, our vein network, the rivers of the world, everything's based on this growth pattern. And the fungal mycelium uses that same fractal geometry in the way it feeds and in the way it moves forward, very similar to how these amoebae organize themselves to construct their giant feeding cell, their plasmodium. So it's obviously one of the most efficient structures upon which many designs in the universe rely on. And we can tap into that intelligence inherently designed in creation in order to help us figure out a lot of problems and solve a lot of issues and make the most efficient systems based on these biological models. There are 4 different sporocarp types of fruiting bodies into which the plasmodial or acellular slime molds fit into. And Physarum, which we have just covered in detail, belongs to a group of slime molds where the fruiting bodies or sporocarps are called plasmodiocarp. These are usually sessile, so there's no stalk involved. They just hug the substrate and they take form of the plasmodial veins from which they were derived. And an example of this is the pretzel slime mold pictured here, called Hemitrichia serpula. This is extremely small in size, so if you ever see a little tiny pretzel growing on a wet log, you will know that this is a slime mold, and you can see how small it is based on the tiny bryophytes growing in the background. So this is what plasmodiocarps look like. It's worth mentioning that there is a wide variety in the size of various fruiting bodies or sporocarps. Some of them are quite small, so you may not be able to notice them easily in nature. So just keep in mind that what we're seeing in the images has no scale, and sometimes these guys are not very obvious as a result in nature. So when you stumble across any of these, they will vary in size depending on the species and the sporocarp that it produces. Another group of plasmodial slime molds form a fruiting body called a sporangium. So while all slime molds form sporangia to disperse their spores, this word is being used here to describe the entire fruiting body or a sporocarp as a sporangium. And this is the most common type of sporing body. So this is some of the way that these organisms look. For example, Stemonitis splendens is a chocolate tube slime mold, which you can see in the image on the right. Hemitrichia caliculata resembles coffee-colored ice cream cones. Trichia favoginea has a honeycomb shape once the spores are released. Arceria denudata resembles pink fairy floss when mature. This is Dacia leucopodia, and of course they are extremely tiny. And you can see why the fruiting bodies are called sporangia, because they have that stalk and that spore dispersal structure. And there's just another image of that again. And the gorgeous lamproderma, which you have seen at the beginning of the lesson. So this is what these tiny little amoebae can do, produce these marvelous, beautiful structures. And they are only available to those that have a macro lens and are willing to look for them in the most obscure places. The next type of sporing body is called athallium, and this is cushion-shaped and sessile and sometimes relatively large. Sessile meaning that there is no stalk, so there's nothing other than an immediate attachment to the substrate. And an example that you can see on the right is called wolf's milk, and these are tiny little balls of Lycogala epidendrum slime mold, which is called wolf's milk colloquially, and they grow on wood and they're extremely beautiful, very tiny. So when you see little bubblegum shaped balls on wet wood, you will know what we're talking about here. And if you actually squish them, this usually pink, very viscous liquid pours out. And this is perhaps why it gets the name wolf's milk, even though I doubt that wolf's milk is actually pink or orange in color. This is what it looks like, and here it's orange, so I think this is where it's now popped by itself and it's actually exuding the spores in this slimy liquid. Fuligo septica, called dog's vomit colloquially, grows on wood chip and mulch, and you'll often see that in your garden, and this is it here. So oftentimes you'll see these masses, and mind you, all of these look very different to what I'm showing in the photos here because they actually change with time. So you might find that at the end of its life cycle, when it's kind of grayish and strange looking, depleted in color, or really vibrant and new and just forming these gorgeous little structures. But this is what dog's vomit looks like. Fuligo septica. The next type of fruiting body is called pseudoathallium, and this is composed of closely crowded sporangia, so the little containers of spores. And once again, it is usually sessile, but few of these might be stalked, so they might actually produce a stalk. Tubifera ferruginosa is attractive, bright coral pink, and it is often called red raspberry slime in the US. It can be pink or red in color, depending on its stage of growth. And once again, if you look up the images of this particular species, they will display various stages of development and ages, so therefore the shape and color changes over time. So this is the end of lesson 6.6. And in our next lesson, we'll look at soil protist symbiosis. So the different relationships that soil protists have with one another, such is the case with the cellular slime molds, but also with other organisms, in the example of algae being incorporated into mixotrophic protists and living endosymbiotically, and parasitic protists that live partly in soil and partly in the bodies of animals. So you'll be hearing from me again in lesson 6.7.